Hi, I'm Violet. I'm a PhD student with the Alliance based in Arusha, Tanzania. I'll be presenting today with my colleagues Hannah Gajasova and Mina Mjema about a large-scale on-farm trial we're conducting in Tanzania this year. The theme is scalable on-farm phenotyping, engaging rural youth for digital data collection. On-farm breeding is important because seeds bred on and around research stations tend to underperform in farmer production environments, which might look very different from the research station conditions. And on top of that, even if they do perform well, farmers may not adopt seeds if they don't meet their specific production and consumption preferences. So there's this real need to get high-quality quantitative crop performance data from farmer fields, as well as in-depth farmer feedback from a diversity of farmers in order to inform breeding decisions and develop locally adapted seeds that people will use. However, this presents a serious logistical challenge. How do we expand the breadth of data we collect without losing depth and quality? In other words, how can we collect this kind of data from decentralized on-farm trials? So how are we solving it? We're configuring a model for a centrally managed study with a decentralized network of farm sites managed by enumerators who are specifically recruited from within each study village where they are living. This design allows us to really make sure in a systematic way that we capture the relevant diversity in farmer and farm types. So to get started, we wanted to make sure we included a range of different environments, and more specifically, environments that are different from each other in ways that are relevant to the performance of the crop we're studying. So for this study, we turned to the bean ecophysiology literature and selected a set of climate and soil variables, that are important for bean crop performance. We threw these into a clustering algorithm and produced this map that basically shows the agroecological zones specifically for beans, and the black lines overlaid show the areas where bean production is high. The ultimate goal is to run on-farm breeding trials in each of these environments. For now, we honed in on the green and red areas, with green being most favorable and red being most drought-prone, and selected 6 districts that encompass this climate variability within the bean production hubs. So here's an example. Within each district, we started with all the villages we could find in Google Maps, here, and we randomly selected 10 across the green and red climates. At several points, people warned us that some of these villages were too far, with bad roads, and that we should replace them with more convenient options. But our decentralized enumerator model, which means we only visit each village location one time, means we're able to include these more remote areas, which are often excluded from this type of data collection, resulting in a convenience bias. This was not without challenges, and my colleague Mina will give you an illustration.
Hi guys, my name is Mina. So today I'm going to share you a story of the challenges we faced getting to the field. So here is one of the story. One day we were getting to the field, a distance of 35 kilometers. You can't imagine we used 6 hours getting to the field. But also we got stuck 4 times on the way getting to the field. You can see this is the first time we got stuck to use the jacket. Didn't help. Here is where tractor came and pulled us out, but you can also see I getting out of the window because now the car door is blocked by the mud. And because we are happy and motivated team in the field, here we are back and we are successfully visited all the villages as planned. Thank you.
Just as our priority was to capture all different kinds of environments across the villages we selected, within each village, our priority was to give all types of farmers a chance to participate, instead of just working with those who are already engaged with extension services or familiar with village leaders. To accomplish this, we invited all bean farmers in a village to a village meeting where we explained the project and collected some basic data using this computer vision-enabled paper survey that allowed us to quickly verify that the selected farmers were similar to the population of their respective villages along some key demographic parameters. So that's where we're at now, planning to begin planting in March, and I'll let Hannah tell you about some of the exciting data we're planning to collect.
Hi everyone! The timeline of this project is 6 months. During this time, we will have 6 data collections. 2 of them will be dedicated to taking pictures for Artemis project, another 2 will take audio recordings for Ndizi, and the last session will be about communicating the result of the study back to the farmers. This farmer feedback session is important for engagement with the farmers and also for them to understand the relevance of their contribution to the study. The project is run by CIAT, and in the project, we are partnering with another NGO called Helvetas that is helping us with data collection in 2 out of the 4 regions. Violet is the main designer of the study and the project lead, and my role is to ensure that operations and logistics are in place so the trials can be executed in the most effective way. Mina and Hassan are at the forefront of farmers' engagement. They each manage 20 enumerators, and each enumerator is responsible for data collection from 12 farms. So altogether, 480 farms. In terms of the data that we will be collecting, this is the first time that we will be collecting data from on-farm outside of research station for the Artemis project. The data will be collected by trained enumerators, and we will be specifically collecting images of pod count and stand count. Ndizi, which is the project to develop speech-to-text AI models for Swahili language to analyze farmers' preferences. We'll be also looking at using the recordings for augmenting tricot preferences ranking data with open-ended responses that we will get. We are opening the study to other projects too. Accelerate will participate via participation of traders in the feedback sessions, allowing for communication between the farmers and the traders and vice versa. Other PhD and master students are also using the data and participating to gain insights for the thesis. So what is the real advantage of on-farm variety trials in this particular case? With the Artemis technology, we are unlocking the capacity to collect quantitative data from anywhere in the world. And if we combine this with the farmer feedback that we will be receiving through the audio recordings, we will really have a foundation for making nuanced, locally targeted breeding decisions that will help us to develop seeds that are locally adapted and that are really responding to farmers' needs. And I would like to leave you with this question for further discussion. How can we overcome convenience sampling bias without compromising data quality and depth? Thank you.